Agents of evolution. We're going to look at the following ways in which changes occur in populations over time: mutations, genetic drift, gene flow, non-random mating, and natural selection. We're going to compare natural selection to artificial selection and how humans, by using artificial selection, are also agents of evolution or changing organisms over time. Mutations are permanent genetic changes. This is one way that new genes are brought into a population. A sudden, dramatic change in genetic information can lead to alterations in the characteristics of the species. Earlier, when we studied genetics, we looked at different ways that mutations can occur. Without mutations, there would not be new inheritable genetic diversity. Mutations are random events. Some are beneficial and some are harmful. Genetic drift is another agent of evolution. Genetic drift is a change in allele frequencies of a gene pool due to random meeting of gametes and fertilization. The random selection and assortment of gametes in a population causes allele frequencies to shift in each generation. Populations are always undergoing genetic drift. Genetic drift has greater effects in smaller populations due to fewer gametes assorting. The removal of gametes from a smaller population can affect allele frequencies in the next generation. If we're looking at this concept of genetic drift, and we have a population of frogs, and a natural disaster kills over half of the green frogs, in this former situation here on the top, this darker frog was only 10% of the population. After the natural disaster, it becomes 20% of the population. So that's going to change the allele frequency. In small populations, we have two different types of genetic drift: the bottleneck effect and the founder effect. The bottleneck effect occurs following a natural disaster that kills a large portion of the population. We just saw this in the example with the frogs. This is a severe reduction in the gene pool that can affect allele frequencies. The founder effect occurs when a few individuals form a colony and their collective genes represent only a fraction of the original gene pool. You can probably think of good examples in human history and the founder effect. So an example of the bottleneck effect: if we have these different marbles representing different allele frequencies, through a natural disaster or human interference, there's only a small portion of these individuals within that population that survive. Then we see how these allele frequencies change. And in this example, we have one marble representing 10 alleles. So notice here how beforehand we had these yellow marbles representing 8% of the total alleles. And if none of those made it through, they're not even represented in the second example. And we can see how proportions of the red, green, and blue alleles have significantly changed. Gene flow is another agent of evolution. Gene flow is the movement of alleles between populations. If individuals or their gametes migrate from one population to another and breed in the new population. For example, wind blows pollen from one population to another, or you have individuals, such as within a pride of lions, that migrate from one population to another, and this gene flow mixes genetic diversity. The gene flow also keeps the gene pools of two or more populations more similar. If we're looking at populations of beetles and we have individuals that migrate to a new population and then breed, and their gametes are migrating, this is going to make this population more similar over time to this one, particularly if we have gene flow between these two populations. Non-random mating occurs when individuals are selective about choosing a mate with a preferred trait. So we see this in human populations and the animal and plant world as well, where there is some trait that makes a mate more desirable. And so random mating is never observed in natural populations because most sexually reproducing organisms select mates based on some trait. So for example, birds with brighter plumage or males that are stronger tend to reproduce more. Inbreeding is an example of non-random mating as well, and this increases frequency of recessive abnormalities in humans. We saw this earlier with closely related individuals in which recessive traits show up more frequently. Natural selection is the last agent of evolution we're going to look at. And as a review, this is a process that allows some individuals with an advantage over others to produce more offspring. That was differential reproductive success. Charles Darwin explained evolution through natural selection. Evolution by natural selection requires 4 events. 1. Individual variation, meaning that there is variation within a population, and that variation is genetic. 2. Inheritance, that variation can be inherited or passed on from generation to generation. 3. Overproduction, this being that there are more individuals that are produced than can survive, and this leads to competition. And 4. Differential reproductive success. If we're looking at natural selection, it's useful to compare artificial selection. Humans are also agents of evolution. They can artificially modify desired traits in plants and animals by selecting to breed individuals with preferred traits. Artificial selection is a type of human-controlled breeding to increase the frequency of desired traits. Both artificial and natural selection are possible because the original population exhibits a variety of characteristics, and this allows humans to select preferred traits. A good example is looking at dog breeds. All breeds are descendants of the wolf, but artificial selection over time has led to the existence of many different dog breeds. Darwin concluded that if humans could create such variety of organisms by artificial selection, then natural selection could also produce diversity. So there was a variation of traits in original wolf populations, and over time, as wolves became domesticated and humans selectively bred for different traits, different breeds of dog were created. There's a Boston Terrier, an Irish Wolfhound. We can look at in the gray wolf and how its lineage has been traced to different dog breeds in Europe, North America, China, and India. Our last concept to look at is that of speciation. A species is defined as a group of organisms capable of interbreeding and that are isolated reproductively from other species. Groups of the same species can exchange genes, but different species do not exchange genes. Mechanisms exist to reproductively isolate similar species. Speciation occurs when one species gives rise to 2 species, and reproductive isolation develops between 2 groups that formerly interbred. So speciation is this development of a new species. A good example are salamanders in California. So we have members of a northern ancestral population that migrated southward, and subspecies are separated by California's Central Valley. Some interbreeding between populations does occur, but evolution, or this change over time, has occurred, and in the south we have 2 subspecies look quite different from one another, and if we're comparing these species to the original species, there's a lot of difference there, and there's no interbreeding between these populations that are further south, and so there's reproductive isolation. Another example is the deer family in North America. This includes mule deer, white-tailed deer, elk, moose, etc. Each one is a separate species. Many biologists believe that all deer in North America started out as one species, and through a process called adaptive radiation, new species arose from the original species due to isolation in new environments over time.